Hey, Nicole Mashburn again, and I want to take a few minutes to talk to you about cell division. Now, when I mention cell division, mitosis throughout some of my lectures, I have to assume that you guys know what I'm talking about because I have to assume that you guys have had some biology. The reality is many of you have not had biology, or it's been years since you've had high school biology. So I'm going to take a few minutes to review what cell division means. If this is brand new to you, you can go into your textbook and do some more reading and go online and learn more about it because you do need to have a basic understanding of cell division. So the first thing we think about cell division, we think about the cell. Remember, a cell is the basic unit of life. It can do everything that it needs to do. It can reproduce. And so the life cycle of a cell includes kind of two main portions. You have what's called interphase, and that's when the cell's growing and doing its job, okay? And then a cell will need to go through the what's called cell division, and this is the mitotic phase. This is when one cell becomes two, okay? So you just make a copy of the cell. And so your cell cycle is basically, you're just gonna think about a circle. So the cell's alive, doing its thing, doing its thing, doing its thing, doing its thing. Then it goes through mitosis, and then you have two new cells. Okay, so that's basically what we're talking about. So again, if this is review, then you're fine. If it's not, you probably want to look in your textbook and read a little bit more about this. But there's some defined events in cell division, and we're going to talk mostly about mitosis. There's also something called meiosis I'll mention toward the end of the lecture. But mitosis, the first step that you have to do when you undergo cell division, you have to divide the nucleus. And this includes four different stages: prophase, metaphase, anaphase, and telophase. Now, you do not have to know what's going on in these different steps, okay? If you're interested, please, by all means, go read about it. But we're not going to test you on the steps going on in prophase, metaphase, anaphase, and telophase for this class. But once this nuclear division has occurred, then you have to split the actual cell. You have to split the cytoplasm, and that's called cytokinesis. So step one is to basically make a copy of your DNA, and then put one copy and kind of split it toward each side of the cell, and then split the cytoplasm in two, and you get two copies of the cell. So you have a cell, you have your DNA, so then you go through mitosis where you wind up with two copies of the DNA, and then you split it through cytokinesis. Okay, that's basically what's going on. Again, don't get caught up in the details unless you're just really interested. All right, so in order to go through the cell cycle and go through mitosis and then split your cell, you have to make a copy of the DNA. And the word for that is DNA replication. So prior to dividing the cell makes a copy of its DNA. Because what you want to occur is if you have this cell with all its DNA, you don't want to have a cell that only has half the amount of DNA in mitosis. Now, in meiosis, that is the case, but for mitosis, you want an equal amount of DNA in your daughter cells. If you have a bunch of DNA here, you want the same amount of DNA in your daughter cells, okay? So in order to do this, you need two enzymes, DNA helicase and DNA polymerase. Remember, ASE tells you it's an enzyme. So the DNA helicase, it separates the DNA strands. Remember, DNA is two strands and they're kind of stuck together. You want to separate that out so that you can make a copy of it. The polymerase is what actually makes the copy. All right, so what does this look like? So here's your chromosome, which is where we store our DNA inside of our nucleus, okay? And it's all wound up in a double helix. And so here comes your enzyme, helicase, and it's going to unwind the DNA and separate it out into two separate strands. Then you're going to have an enzyme called DNA polymerase, and there's going to be one on each strand, okay, because you're going to copy both strands, and it's going to come along and it's going to make a copy of the DNA. Now, remember back when we talked about DNA and we talked about organic chemistry, and I told you about adenine, thymine, cytosine, and guanine? These are the bases that we use to make the DNA, and they're paired. A and T always go together, and G and C always go together. So you can see here is thymine, and here is adenine, and here is cytosine, and here's guanine, and A and T always pair, and G and C always pair. So what happens is the DNA polymerase finds the base that it needs to match, and it puts the corresponding matching base there and makes a copy. So now you've copied this strand, this is now your copy, and here's your original strand, and you're making a copy. Okay, so that's DNA replication. You take your DNA, You divide the strands using an enzyme called helicase. Then you use a polymerase, poly meaning putting together multiples, and then you add the corresponding bases, the matching bases, and make another strand. Okay, again, you're welcome to read more about that, but this is really all I want you to know is that DNA replication is how you copy your DNA. So in DNA replication, the end result is you get two identical DNA molecules formed from the original DNA. So in mitosis, when the cell divides, one copy goes to the new cell and one stays in the original cell. So you take this cell, you double the DNA, and then when you split it, the original cell keeps its original copy, and then the new cell has a copy. Okay, this is called semi-conservative replication. Just want to throw that out there in case you've heard that. And what that means is each DNA has one old and one new strand. So remember when you had the two strands and they split apart and you made the copy, one of these strands has a new and an old, that goes to a cell. The other strand has a new copy and the old copy, and it goes to the other cell. Okay, semi-conservative conservative replication. Now, once you've replicated your DNA, then you can divide your cells. And there's two types of cell division. We've been talking about mitosis, but there's also something called meiosis. Now, let's talk about mitosis first. Mitosis is when you produce a clone. You make two daughter cells that are identical. This is what our body does all day long. This is how you make bodies grow. This is how you make bigger bones, more skin, more blood cells, how you repair tissues. Mitosis is what's going on in all the organs of our body all the time. It's always occurring in your skin, your intestinal lining. You're making more fat cells. So mitosis is how you divide and make copies of more cells. Now you don't see this in mature nervous tissue or mature skeletal tissue or cardiac tissue. Okay, those types of cells do not undergo mitosis. Once those cells are mature, that's all you got. You don't make any more. Now, meiosis is different. This occurs in the reproductive organs, in the ovaries of the female and the testes of the male. And this is how you make the gametes, or sperm and egg. And so meiosis is a little different. You do copy your DNA, but when you copy the DNA, okay, it's going to eventually divide and produce actually four cells, four daughter cells. And each one of these daughter cells is going to have half the number or half the amount of DNA in it. So meiosis produces daughter cells with half the genetic material, which is a good thing. So if you get an egg from your mom with half the amount of DNA that you need, and then a sperm from your dad with half the amount of DNA that you need, you put those together, then now you have all your DNA. Okay, again, this is very simplistic. We're going to cover this again when we talk about reproduction. I just want you to know basically what cell division means so that when we do mention it, because a lot of times we forget, we mention it casually and we say meiosis or mitosis, assuming that you know what we're talking about. So hopefully this will help you review. And again, you're welcome to watch more videos on the internet or go to Mastering, go into your textbook and learn more about it. That's all you need to know right now. Thanks.